In this video, we will go over the project we started in 2017, the Common Orchid Project. This is a project we are doing in collaboration with the North American Orchid Conservation Center, NAOC. Who is NAOC? They are the North American Orchid Conservation Center, a coalition of organizations dedicated to conserving the diverse orchid heritage of the United States and Canada. Based on the Smithsonian Environmental Research Center, SERC, it also includes a list of collaborating organizations that is growing. With representation from most regions of the U.S., NAOC has also developed an interactive website, Go Orchids, to provide the public with a mechanism to identify and learn about our native orchids. You can find a link to the website on the Moodle course. The goal of the Common Orchids Project and our collaboration with NAOC is to better understand the conservation and propagation of our native orchids. We hope to better understand native orchids by collecting seed for seed banking, leaf tissue for DNA studies, and root tissue to preserve and propagate mycorrhizal fungi association with orchids. These collections are ideally done by ecoregion. However, we have created target species lists by state because we want to avoid collecting from federally listed and state-listed species, including those species on watch lists. We have created a state-by-state list of orchid species we are targeting, so please check your state list to learn about what species we are looking for in your state. 2017 was the first trial run year, and we learned a lot. All collections in New England are coordinated through Native Plant Trust, and, similar to our rare species surveys and seed collections, landowner permission needs to be obtained first before we can pursue a collection. We encourage all of our volunteers to let us know when they have found common orchid species, whether that was during a rare plant survey or when they were out hiking. In 2017, we completed 15 collections, 7 in Massachusetts, 4 in Maine, 3 in Vermont, and 1 in New Hampshire. All of these collections were completed thanks to volunteers reporting what they found, and then working with us to complete the collections. Some collections were done on public lands, others on private, including a few volunteers on their own properties. Here's an example of a collection completed by the Conservation Department staff in Massachusetts, Goodyeana pubescens. You can see this is a really nice and big population of this species. About 100 rosettes were found, and about 40 flowering stems. The picture to the left shows the plants and flowers, and the picture to the right shows the plant and fruit. This is what a leaf tissue sample and a root tissue sample look like in their approximate sizes. From this population of about 100 rosettes, 5 collections were made of root, leaf, and seed from 5 plants. Because certain species of orchids can grow clonally, where some of the rosettes are the same genetic individual connected by underground rhizomes or stems, such as Goodyeana pubescens, we were careful to sample from 5 individuals at different sections of this patch. As with most orchids, the seeds are like dust for Goodyeana pubescens, shown in these photos. Seeds, roots, and leaf material were collected from the same plant, making sure not to disturb the plant too much in taking these samples. Only 5 collections were made at the population to ensure we did no harm to the population itself. Going back to the logic of the sampling of orchids, here is a picture of the 5 ecoregions found in New England: 58, Northeastern Highlands; 59, Northeastern Coastal Zone; 82, Acadian Plains and Hills; 83, Eastern Great Lakes Lowlands; and 84, Atlantic Coastal Pine Barrens on the Cape and Islands. In each state, we want to collect from each ecoregion found there. For example, for Begonia ophioglossoides, we want a collection from Vermont, New Hampshire, and Maine in the northeastern highlands, depicted in purple in this map. Also in Maine, we want a collection from another ecoregion found there, the Acadian Plains and Hills. Maine is the only state where the Acadian Plains and Hills ecoregion is found in New England. And here is another depiction showing the collections we want from each state and each ecoregion. For New England as a whole, we are looking for 14 total collections per species. Again, we are aiming to collect by state and then by ecoregion in each state. Collections are to be completed as best as you can by maternal line collections, meaning all of the material should be collected from the same plant. Just like for rare plant seed collections, we only want to collect 10% of seed from any individual in the population, 1 leaf and 1 root as well. If you are not able to do a maternal line collection, contact Native Plant Trust or NAOC to see if you can still send in samples. In the past, we have accepted samples that were not maternal line collected. We encourage the collection trip to happen in the summer or autumn to collect materials. Please make sure you review the protocols provided, and as we further refine our methods, new protocols will be passed on to you. Please provide us with any feedback and information about how to clarify and improve the protocols. If you do any collections, NAOC and CERC ask that you sign and return a collector's agreement. Landowner agreement forms, in most cases, are needed, and Native Plant Trust will help in getting these filled out. We also need to make sure any collection permits from the state or landowner are filled out as well. Something again that Native Plant Trust conservation staff will assist with. It is absolutely essential that you completely fill out the field form for your collection and properly label your collections. Instructions on collection labels are outlined in the protocols and right on the field forms themselves. Some tips on how to collect and recognize when to complete your collections. As with any collection, sometimes it will take more than one visit to the population to determine the best collection date, especially for seeds. For fruit and seeds materials, they are best collected when the fruits have almost matured and begun to yellow. These are best left to dry completely after a collection. Orchid seed is like dust. Please do not collect or store seed or dry pods in plastic bags. The static charge makes it hard to recover seeds. Instead, either use the glassine envelopes we use for rare plant seed collections or coin envelopes. Leaf material and roots should also be collected at the same time of seed collection. The leaf collection for DNA material can be dried with a desiccant such as silica gel or beads, or air dried in a coffee filter and placed in a coin envelope. For plants with large leaves, half or a quarter of a leaf would be enough. Seed should be sent to Nasami Farm Nursery, the same place we have volunteers mail rare plant seed collections, as soon as a collection is made. Nasami Farm Nursery, Nepcob Seed Bank, attention Kate Wellspring, 128 North Street, Whately, Massachusetts, 01373. Leaf materials and root materials should be sent together to the Smithsonian Environmental Research Institute. Attention J. O'Neill, Cirque, 647 Contees Wharf Road, Edgewater, Maryland, 21037. Here are a few tips and facts about orchid roots and associated mycorrhizal fungi. The roots are collected for fungal studies and propagation of these fungi, and are best isolated and grown from fresh root samples. To determine if your roots have a good fungal infection, roots that appear slightly yellow generally indicate this, and are better than the new white roots. Please do not dig up the entire plant, and be careful when sampling the roots as to not disturb the plant too much. The photo here is just an example of what the roots look like, and is actually a propagated orchid from Garden in the Woods. Please collect only one root, and the entire root piece up to the plant. And again, be gentle and careful as you collect the root, only minimally disturbing the soil and making sure to cover up the area you collected from. When you collect a root, they will need to be cleaned and wrapped in a slightly moist paper towel and then placed in a Ziploc bag to keep them moist. The best time of year to collect roots varies as infection of the mycorrhizal fungi. However, for our purposes, we will collect roots when we collect seed, sometimes in the summer or autumn, depending on the species and when it goes to seed. Root materials and leaf materials need to be sent to the Smithsonian Environmental Research Institute, attention J. O'Neill, Cirque, 647 Contees Wharf Road, Edgewater, Maryland, 21037. Please send your root and leaf materials as quickly as you possibly can. Expedite or overnight shipment preferred to ensure that the fungi on the roots survive and they can isolate and propagate the fungi. Please also email Joe O'Neill when you ship your roots so he knows to expect them and to take care of them as soon as they arrive. For those of you interested in participating in the project, please keep an eye out for common orchid species and be sure to report them to us. Here is what the Common Orchid Sighting Reporting Form looks like, and we need your help in determining where populations of common orchid species are. If you are interested in doing collections but have not found orchid populations, let us know. We might be able to have you help out by scouting areas we think orchids might be located and completing collections if found. If you have any questions about the project, please contact either Micah Jasny, Botanical Coordinator at Native Plant Trust, or Julian McGinnis, the NAOC Program Development Coordinator. We hope you can help out this season in the Common Orchid Project. Thank you very much.